Yeah, before I start, I'll just say a little bit that I won't say a lot about how we solve any of these problems, but we also have astrophysicists, computational chemists, computational biologists all working in the same building. So hopefully over the course of the day, we can talk about how to put those things together. My lab works on a lot of different topics. So we work on approximate network inference. We work on Bayesian inference for genomics data, and we work on protein structure. And today I'm going to talk mostly about just the latter, the protein structure part of things. And the goal is to actually describe some work in machine learning that is inspired by the actual structure of the biophysics. So we want to use protein structure to extract function from protein sequences, to predict the function of proteins from protein sequences, but we want to do it in a way that we still maintain some hope of understanding the mechanism. And so this will be a lot about the machine learning because that's the part that's new for us, but I'll talk a little bit about the older ways of generating protein structure. All right, so because we might have non-biologists in the audience, this is the problem. This is the start of the problem. So because of sequencing of DNA, because the technologies for sequencing DNA have their own super Moore's law. So it's like Moore's law, but the exponent is a few months shorter. Because of that, we actually now have 100,000 genomes if you ask Rob Knight at UC San Diego, or 10,000 genomes if you ask someone slightly less crazy than him. So if you talk to people who sequence DNA from the environment, they say we have 100,000 genomes. If you look at the genomes that are carefully annotated, we have many fewer. But we know that we actually have millions of these protein sequences sitting around, and most of them are unannotated. But those protein sequences contain new enzymes, they contain proteins that mimic proteins in the immune system, they contain targets and pathogens, and they contain many, many, many unannotated proteins. So this sequence that I showed you, if you cross your eyes, you can see in 3D here. So this is P53 bound to DNA, and this is the protein that is mutated in 80% of human cancers. So this is a very important protein. Another way to look at this is as a tree of life. And so we have some last universal common ancestor of life, we believe, that then sprang into the eukaryotes and the prokaryotes, the things with nuclei, the things without. In this vast number of sequenced genomes and parts of sequenced genomes from environmental sequencing, we have a huge amount of biodiversity. And there are lots and lots of applications. But today, I'm going to focus on the process of sifting through those sequences. I'm going to show you one method we're going to develop. But in general, as sequences, the number of sequences has increased, there's not just the challenge of annotating those sequences, but many of the methods that we use to annotate those sequences benefit from having more comparisons to make. And so in general, if we look at the tasks we might need to do, we might want to predict the structure of those proteins. We might want to predict the function. These are the two things I'll talk about today. We also, though, if we're dealing with proteins that are in our crops or our bodies or the pathogens we care about, we might want to know whether or not variants affect the function of proteins. So single changes in the sequence, we might care if those affect the function of proteins or their interactions with drugs. In general, there's a huge expansion in industrial biosynthetic capabilities. So actually, Merck, for example, has enzymes involved in the processing and the generation of most of their drugs. And of course, I mean the Merck in the U.S., not the German Merck, sorry. I don't know as much about the German Merck. All right, in any case, today I'm going to focus on this part here, and I'm going to try to show you that using predictions of structure, the three-dimensional structure of folded proteins, we can do better at predicting the function of those proteins. And so before I go into that, I'm going to just go through what I mean when I predict protein function. And so if we just take a brief pause to consider how many protein functions there are, even if we're not biologists, we know that life does quite a bit, right? So if we just think of biosynthetic the generation of biomass, there's a huge number of molecules that have to be synthesized with enzymes. So those things all have to be functions. We know that life has to regulate its own growth and development, so there's lots of regulatory and informational processes, and the list goes on and on. And so in order to encapsulate the function of proteins, biologists and computational biologists over the years have built many different systems. One system is the gene ontology system. Another is the enzyme classification system, the EC system, which is encapsulated in a database called Brenda, and the list goes on and on. There are probably 4 or 5 that are widely used, and today we're going to focus on the gene ontology because this is a system that encapsulates lots of prokaryotic as well as eukaryotic. Many systems are better for human, but they don't work so well in bacteria. Some systems are actually very, very tuned towards enzymatic synthesis and the things that people focus on when they study bacteria. And Go is something that encapsulates a great number of different organisms. So it's the most widely adopted. In general, the way all of these systems work is that they start with general processes and they descend to more specific functions. And so if this is a protein, this is actually the tree for interleukin 17, which is a protein that is important for inflammation. And so it is something that regulates cytokine activity by binding the interleukin 17 receptor, which then has a number of other parents. These are the functions that actually matter, and then these are the things that are in place so that the ontology is complete. Why is it an ontology? Why does it matter? There's a number of reasons for having this thing represented as a tree, but in general, this is what we're going to try to predict, and there's multiple trees. So there's a function tree, which essentially tells us what are the parts. There's a biological process tree, which tells us what pathways you participate in, and there's a cellular component tree, which tells you where in the cell physically is the protein localized, since localization of proteins is important. If we look in this particular database and we look at Homo sapiens, we see that actually there are about 200,000 annotations that have experimental validation and many more that are inferred electronically or inferred through multiple different methods, mostly through very strong sequence homology with mouse, rat, chimp, et cetera, et cetera. If we look at Gallus chicken, we see that almost everything comes from comparison to another species. If we look at rice, if we look at any of our crop species, then it gets much worse. So as we go away from the most widely studied organisms like S. cerevisiae and Homo sapiens, we actually see that there are many fewer annotations and that most proteins are of unknown function. Now, if you're of unknown function, we might know that you bind a receptor or that you regulate signaling or that you're a transcription factor, but we might need to know more than that. We might need to know what part of the protein does the function, and we might need to know in proteins with the structure of multiple domains, which domains do that. So in addition to figuring out who has what function, we also want to map that function to different specific parts of the gene in the protein because that's the heart of mechanism. All right, so the last piece of the puzzle is that I showed you that large evolutionary tree. One thing that complicates judging protein function or judging many things in computation is that all of the parts of these systems have evolved from this last common universal ancestor. And so there's often ways in which people fool themselves. They think that they're doing much better than they are because they're mapping more through evolution, through comparison than they think. And so it's very important in studying protein function, structure, and all of these other sorts of things to have a proper blind challenge, to have a proper community-wide blind challenge. And so this paper here describes the CAFA competition. So every 2 years, they release which organisms they're going to focus on, and the challenge is to predict all of the unknown proteins in usually around 20 organisms. They wait 1 year for new annotations to accumulate, and then groups are judged. Methods, I guess, not groups, are judged based on the temporal holdout. And so this is something where we as a community try to predict proteins of unknown function, and then the validation set is every single functional annotation that's deposited in the intervening year. This is an early version of that. This was mus musculus, which is mouse. This is the lab strain of mouse, and this was done many years ago by Fritz Roth and Olga Tryanskaya and a bunch of other people. And this temporal holdout is the difference between knowing if it works versus, having a lot of uncertainty as to what you've mapped via evolution. Okay, so everything that I'll describe today, all the numbers are going to be from a holdout comparing inputs from 2015 and then validating on annotations that were deposited in 2017. So we have a 2-year temporal holdout for everything that I show you today. Overall, when we try to predict protein function, the last sort of thing to consider is when you make a prediction about protein function, what's the basis for that function prediction? And I think these are the 3 major types of inputs. And again, I think there's a few people who are not biologists. Hopefully being here, you know a little biology, but I'll describe these things a little bit. So protein networks. There are a number of ways in which proteins or genes can interact. Some are specific to proteins. If proteins interact with each other, then we would put an edge between those 2 nodes. There's also coevolution and a number of other interactions between proteins that are actually a feature of the system or of the evolution of all the genomes that we know of. So co-interaction, coevolution, and relationships from context in the genome are all potential interactions here. Some of these are actually specific to branches of life. So in bacteria, operons, being in the same operon or being very close in the genome has a much stronger functional significance than if you're close in the genome in a eukaryote or a less gene-dense organism. These are the 2 things that I'll talk about today most of all. And so this is a diagram that shows the sequence we might be interested in at the top, and underneath it are aligned many variants of those sequences found in other organisms, or in some cases, different versions of that protein found in the same organism. So this is called the multiple sequence alignment, and we can think of it as just a large matrix that shows us for the length of the protein, what other sequence positions are possible. We can also think of it as reduced to the length of the protein times the frequency of the 20 amino acids at each position. So both of those things can be direct inputs into machine learning algorithms and to other things like algorithms that fold proteins. The last part of the puzzle, or the last potential feature, is protein structure. And here we can take this sequence. These letters indicate different amino acids, and the folded structure has a great deal of functional information in it. In general, this was the first type of data that was used to predict protein function. In the very early days of genomics, almost all functional annotation was done by mapping protein functions from experimentally understood proteins to others via very carefully curated, typically hand-curated multiple sequence alignments. Eventually, this was codified in lots of programs that looked at orthology and other more complex evolutionary relationships. In the sort of mid to late 90s, we saw a bunch of experiments that used guilt by association and network diffusion and other models that sort of had their, that were born in the stock market, essentially. So network diffusion models and other ways of integrating multiple networks were used to transmit function between known and unknown proteins. The best example of this is BLAST, I guess, the most widely used program. The best example here is a program called Gene Mania by Sarah Mustafavi. She did that when she was with Quade Morris. Getting functional information directly from structure is something that we've seen less of. Many people have done things where structure is taken from structure, function is predicted from structure, but many of those things involve lots of feature engineering. One of my favorite examples there is by Andy Wallace, who, when he was with Janet Thornton, they built a database of active sites for, I think, around 40 functions, and then showed that they could search the protein data bank and find cases where annotated proteins had additional functions. When they tested them in the lab, they actually found that many of those discovered active sites were real. And so that was kind of an interesting way of showing us that just because you know a protein has a function, it might have other functions grafted onto it by evolution. And so that was one of the first cases where I saw some nice use of structure. In any case, this is something that's often wrong. This is something where guilt by association ignores many of the things that happen in evolution, and this is something that's difficult because it's not really been done automatically before, and it's not always easy to get structures. In fact, it's never easy to get structures. All right, I'm just going to advertise this paper for people who like networks, and then I'm going to move on. This is the thing that we have a nice paper about. Everything that I'm going to talk about now, Vlad is typing away trying to get up on the bioarchive. So deepNF was a way of integrating different networks from different sources. So we might have protein interaction, we might have coevolution, and we might have networks from different species. It was a way of using diffusion models or PageRank models to turn these into features, which we could then use in a self-supervised framework called deep network fusion to make features which we could then use for protein function and other things. And this seemed to beat mashup and some other methods that came out of Bonnie Berger's lab at MIT, and so we were excited about this and published it, and we have new versions of this that take a wider variety of inputs. Okay, so this is one example of how you could automatically deal with networks. Again, that was just an advertisement for that paper. What I want to focus on is how do we take multiple sequence alignments and how do we take multiple sequence alignments and extract function from them, and then from there I'll move on to how do we combine sequence and structure information. All right, and so again, the state of the art in about 10 years ago for sequence information was just that you would take your multiple sequence alignment, you would look at what the thing that you're interested in is. So let's assume that this is a protein that we don't know the function of, even though all of these proteins are of known function. We would then look at whether or not any of the aligned similar sequences had function, and there are many, many different ways that people interpret this. So we might say, I only want to transfer function if I'm your closest match and you're my closest match. We might want to look at the phylogenetic tree and make some inference based on the tree. So there's actually a great deal of good work to make transfer of sequence based on homology, or transfer of function based on homology better, but all of that work essentially boils down to finding sequence similar patterns. A lot of people, including our group, in the last few years have tried to come up with ways of learning function that actually use the sequence directly instead of actually doing transfer by homology. And so I'll show you, just as a way of sort of warming up, this convolutional neural network approach to function prediction, and then I'll get to the thing that we're actually interested in, the structure-based method. So in the end, a convolutional neural network is something that takes multiple regions of the field, and so here this field is a picture, and so we're going to take filters that are essentially square matrices. There'll be some linear filter that then produces a number, and the reason this is a convolutional neural network is because we can do this multiple times, and there are of course many architectures beyond just the convolutional neural network, but the idea is we might have features from one area, features from another, and then a convolution of that first set of filters with another set of filters will allow us to make inferences that involve multiple regions in the picture or multiple sets of rules applied to the same region. As we get to deeper layers in the network, we have abstractions that lead to the final prediction. So the rules, as we look at the filters that are closer to the raw input, we actually have things that are closer to edge detection and other filters that came out of sort of early days of vision. And so this is something that tries to do that in an automatic way without feature engineering. If we're looking at a multiple sequence alignment, we have a number of ways we could represent the sequence. And so first I'll show you, what we would do if we had, if we have a multiple sequence alignment. We could actually look at the number of k-mers. We could one-hot encode the original sequence, so this is the sequence we care about, or we could look at the multiple sequence alignment, and we could say what are the log odds of seeing that particular amino acid, and so this would be alanine, cysteine, da-da-da. So this would be the length of the protein times 20. We might put a 1 if we've got it there. We might put the vector from that column of the multiple sequence alignment. So we have multiple ways we can encode the sequence for a convolutional neural network. And again, I'll just show you kind of the basic idea. For each filter, we might have multiple filters. We have to learn weights for the position I and position I plus 1. And then once we have a filter, we drag it across the whole sequence to make a vector. And the only thing we learned were these particular weights, and we can have constraints on these weights and other ways of regularizing this system. But if we have another filter, we drag that across the sequence. And the idea here is that a real working convolutional neural network will have a great number of these features. Then we'll have filters for the second level, which essentially convolve the first set of features with the second set of features. And off we go until we have a certain number of layers. All right, and here the sequence we have a certain field or perceptive field that's essentially the width of the filters. By the time we get to the nth layer, that perceptive field is widened quite a bit by the convolution of those filters over multiple steps. So there's a limit to how much the sequence, a set of rules, can be applied to in the end, but it's essentially L times the width of the filter. So it's not the length of the sequence, but it's quite a bit longer than just the single filter. So the idea is you still have some ability to learn distal relationships and distal rules. And the last part of this neural network is a neural network that resembles neural networks from the mid 1990s. So these are just dense sets of nodes with linear relationships or truncated linear relationships at every position, and the convolution or the number of layers gives you all sorts of ability to represent essentially complex functions. So this is the structure here. We have in our convolutional neural network, I think we have 6 layers, which is not that deep, and then we have 5 layers in this function prediction layer. We have all the Go terms, so there's 2,800 Go terms, say, if you're in the function tree. So this is a complexity of predicting protein function is that the number of labels that you're simultaneously trying to predict is very high, and there are relationships from the ontology between those labels, so there's some complexity there as well. All right, so if we do that, if we use what we considered the simplest convolutional neural network that we thought could do the job, we got reasonably good performance. And so if we look at just the key parts of this, so this is BLAST. And so these here are things that are essentially based on sequence similarity. So this is the micro AUPRs. This is the area under the precision recall curve if we allow every function to vote once. The macro, this is everything in every genome. And so every protein gets 1 vote here, every function gets 1 vote here. And the reason this is important is because there are some functions that you see 1,000 times in a genome and some that you see 10. And so this is balanced across functions, and this is balanced across membership in the genome. In any case, you see pretty much the same trend in both, which is that these various convolutional neural networks and slight alterations in the input and the convolutional neural network architecture all outperform transfer based on homology. Things based on k-mers, bag of words, things that mix the features and don't take account of the structure of the sequence also don't work nearly as well. In the interim, while we were experimenting with that, a number of papers, I think 3 in 1 month, all kind of came out. This was in sort of mid-2017. So we saw ways of predicting function based on the sequence. And again, here they did a lot of feature engineering work actually by embedding the predictions in the ontology. So that was kind of nice. prediction of localization, and prediction of protein fold. All of these papers kind of came out. There's also been a lot of work for predicting protein structure directly from multiple sequence alignments as well. So we've seen a lot of pure sequence to various things, papers coming out. This group here, actually, there's a number of limitations in terms of what they produced, and so they limited to a very, this paper is actually limited to a very small set of training examples, small set of organisms, and a small set of subfunctions. So there was actually quite a bit of focus, let's call it, in this paper. So this is not what we would consider a generally useful tool, and these things are very specific as well, to just very small sets of labels. All right, so we see good performance of pure convolutional sequence based predictors of protein function, and we have a lot of evidence from other groups that there's a lot there, and that we can, I wouldn't say transcend homology, but we can capture a lot of rules that you don't see with just closest mutual match and other things like that. So the next sort of part of the talk is the actual good part. Sorry for all that introduction. Here I want to talk about how we use structure, and I just want to introduce one small twist to the method, and then I'll show you, essentially spend the rest of the time showing you how we test it and how we put it together as a proper method. Okay, so with convolutional neural networks, we assume some metric space typically. So in biology, we often assume that there's meaning to moving one character in the sequence, whether it's DNA or whether it's protein. In images, they assume all kinds of things. So if you take some group that says they solved the vision problem and then you show it histopathology, if you rotate the image or screw up the normalization of the image, it matters a lot. So there's a lot of cases where things show up in NIPS and they say we solved the vision problem, but as soon as you apply it to anything other than pictures of a human situation with a horizon, they die. Okay, so there's metric space and there's often rotational variance that's problematic. So there's been a huge group of people. Jan LeCun and Johan Bruna at NYU are people that, due to my proximity, I'm biased towards mentioning, but there's a bunch of really good people who have started to think of non-metric spaces, or spaces where the relationship between how the feature space is built is something that might have a lot of variation beyond sort of a human readable space. And one thing that it's very hard for people to process are very large graphs. And so a graph convolutional neural network is a reasonably new architecture that we decided to experiment with. And again, we had neighbors who could help us out thinking about some of these things. And here the idea is, instead of having filters that scan over the image, instead we're going to have filters that scan over the network. And for us today, the network will be an encapsulation of the protein structure. And so here is a protein whose name I've forgotten because it fell off the slide, but there's a great number of these proteins. This is a protein that has 2 or 3 domains, depending on how you were raised to look at protein structures and things like that. But we have essentially the protein folding into this little alpha beta. We have a helical part here, and then we have another helical part here. And so if we draw the graph where each node in the protein is a C alpha carbon, and we draw an edge between those nodes if the distance between those C alphas is less than 10 angstroms, then we get this graph. And this graph is also something that we could draw as a square matrix called the contact map. And so we're going to experiment with convolving over this graph as opposed to the primary sequence. Before we do that, I just want to point out that the prediction of protein structure is also something that has seen a lot of advancements in the last couple of years. So there's a number of methods that actually predict contact maps and then use a program called Rosetta to predict the protein structure of. There's also David Jones has a method that again predicts a contact map and then uses a software that's been around for a while to refine NMR and other experimental structures. And so there's a couple of different pipelines. They all have differences in their parts, but they start off by using very large multiple sequence alignments to predict contact maps based on evolutionary covariance, and then they predict protein structure with lots and lots of constraints to keep you from having to deal with a lot of the sampling issues that you get with protein structure. Okay, so this is something we used the David Jones method because Tomas is part of the team. And so both of these methods, the David Baker method and the David Jones method, were the methods that we use. We put those through Rosetta, and again, that's because that's the code we contribute to, so we're extremely biased there, but Rosetta does all right. De novo structure prediction is the thing we'll use it for today, but it also does fold and enzyme design, protein docking. Most of the licensing money we get from this code comes from people just doing things with antibodies over and over again. Same fold, same function. So it does 1,000 things, but the world apparently only cares about one thing, which is making antibodies tougher. But the way that we are applying this, the goal is that we want to fold all the proteins that are out there, but to do that, there's a big scaling issue. So we've teamed up with IBM to make a screensaver that runs from the World Community Grid project. So we have about 400,000 people in the world who have donated their personal computers to folding these proteins. And this is a sort of typical benchmark. So this is a protein, we folded the entire PDB, and we looked at the quality of the outputs, and this is the median. So this is exactly in the middle. This is not at the top. And essentially we get to within about 2 angstroms. And the thing that's important for today's talk is that when we look at the predicted contacts from things like EVFold or Gremlin, they're very sparse, but they're pretty accurate. And when we actually run them through Rosetta, we build out the rest of the contact map, which is important if you want to diffuse through those maps or use them in the way that we want to use them. We estimate that we're discovering about 50 new folds a day because we're running 100,000 proteins through this thing. That's another talk. And now we have the input for a graph convolutional neural network. That's the key thing here. So now we're going to take these protein predictions and we're going to let the graph convolutional neural network actually build up feature sets that are based on these predicted proteins. And to do that, we're using nearly all of the PDB as a training set and also as a subset of this as a validation set, because a great number of things are deposited into the PDB every year. So in the end, when you look at the protein data bank, there are actually a good number of chains, but when you reduce redundancy and you look at which of them actually have high quality function annotations, it reduces to a pretty small number. We're also working with the people from Swiss Model to actually make this number much bigger, under the sort of using the sort of belief that homology models are good enough for function prediction, right? If we also want to then later say Rosetta models are good enough for function prediction, we definitely have to say that. All right, so the full structure, one thing that I find interesting about these machine learning projects is that these days, due to the volume of papers, due to the fact that there's a lot of good platforms for building these methods, these architectures sometimes get a bit overcomplicated. So let's go through the first thing. So the first part of the input is the protein structure represented by a square contact map. We experimented with a lot of diversity in how we build the contact map. If we have the structure, should we build the contact map based on C-betas, hydrogen bond networks, some of the more extensive details of the structure? Should we have multiple contact maps? We experimented with a number of these things, and what we found was that just the C-alpha contact map was not, we couldn't prove that it was worse than using the more complex multi-view version of the structure, which was depressing to us because we like structure. But in any case, this is the main input. C-alpha 10 angstrom contact map. And so this contact map then goes here, and the convolutions over the sequence are happening over the primary sequence, but the convolutions are allowed to teleport through these contacts. And so That's the part that I've explained, I think, a little bit redundantly at this point. Now, the other part of the puzzle that I'm going to focus on less today, because this is sort of a sequence thing, and I'm less excited about sequence, is that we still have to decide what are the vectors of features that sit on each of the nodes before we do any convolution, right? And so for this, we use a bidirectional LSTM, and so this is a model that essentially was trained by taking 2 million proteins, or all of the proteins that are non-redundant, that are high quality, they're not fragments. These are the sort of large unannotated sets from Interpro. And what you do is you try to predict, you leave out residues and you try to predict residues. And once you get a machinery that can predict hidden residues, then you can use that same machinery to predict vectors that represent not just that amino acid, but its local context. Okay. So that's the language model, and there are a lot of groups that have shown a lot of power using LSTMs, word-to-vec-like models. There's a model called BERT, and I can't remember what BERT stands for, but essentially all of these things are hiding parts of sequences, in this case, the sequence, the primary sequence of amino acids. They were all developed to hide different parts of strings of language and then predict the missing parts. And so this is coming from natural language processing and from translation. So this is an LSTM without an attention mechanism. That gives us what's painted on the nodes, and then the machinery is building up rules that are essentially rules applied to walks through the contact map. Okay, so the first thing to do is just to see if we add this additional information, structure, does it do better than the convolutional neural network? And one thing to keep in mind is that the convolutional neural network is a powerful way of making distal rules. So the worry was that we were going to build this complicated thing, but with enough protein training examples, the convolutional neural net would learn the rules that the structure represents and beat or tie the more complex method. And lucky for Vlad, because he does want to get a faculty job someday, if we look at BLAST in gray, the convolutional neural network based on sequence in blue, and his graph convolutional neural network, the structure-based method in orange, we see that for enzymes, Brenda, this sort of enzyme-dominated database, molecular function branch of Go, and cellular component, his method beats all the other methods that we can find, and in particular beats the comparable CNN that's also using the LSTM features. If we look at biological process, that's what pathway you're involved in. Here we don't do better. The best method that we're able to find, either from our own work or from other, is the convolutional neural network. And there, these are the things that are organismal context. They're less site-specific. And so for us, this actually kind of makes sense that the sequence-based method works better for this more contextual function. If we focus on the function part of this, we also test whether or not the CNN with one hot encoding, the GCN with one hot encoding, this is just the raw sequence, or the language model does better. We do see that the language model painting the nodes does a lot better. Again, we have to see here how well we do, but we also have to look at training examples and test examples and validation examples that have different relationships between them. And so here, what Vlad is plotting is if we build validation sets that have 90% sequence similarity to the training set, does the model learn better or worse? And what's the sort of limiting behavior of performance as we have more distant things? And so if we go down to 30% or less maximum sequence identity between the training set and the test set, we see that the performance actually is still maintained at something that beats BLAST by quite a good margin. All right, and then it is important to note that there are still many things where if you have a 50% sequence identity and you map the function via 50, 70, 90% sequence identity, the function that you map is wrong, right? We have to remember that first plot I showed you, that if you map via BLAST, you get the function prediction problem wrong half the time, even in sequence similarity ranges that are very high. You definitely would have the same structure here, but you don't always have the same function. All right, so this is a plot that's a little bit complicated, but essentially these are all different functions. And then these functions, there are many examples across many organisms. This bar plot here is the AUPR, the area under the performance recall curve, for each guy. And so 1 is perfect, somewhere around 0.05 is random, and these are all functions where the GCN does well and the CNN fails. And so we're ranking here to show some of the functions where we do much better than the sequence-based method. And what Vlad did was he plotted the number of training examples versus the difference. And what we see is that if you have a large number of training examples in the training set, then the CNN can learn those rules. And if you have a smaller number, then the graph convolutional neural network starts to beat this. And so I think this makes a lot of sense in the sense that if you have the protein structure telling you which parts of the sequence should be combined in a single view, then that's one less rule that you need to learn. The CNN can combine things, but it needs more layers and more parameters. And so the number of training examples, if you have 600 versions of that protein, then you can learn these complex rules. But if you have 50, then you need some guidance as to how to build those filters, and the structure provides that as a constraint. The other thing that we looked at, again, if you look at how wide the filters are and how many layers are, you can do lots to find distal relationships between parts of the sequence, but there's a limit. And so as we get to the larger proteins, so this is average length versus the difference in performance of the GCN minus the CNN, again, you see that the GCN does a lot better for the proteins that are larger than 200 amino acids. And again, there aren't, the average size of a single protein domain is in this sort of range here, right? But we often don't know the domain structure and other things like that. And so there are many, many cases where we'll need the predicted structure to actually figure out the relationships. All right, so I'm going to skip over what Rosetta does and just show you that when we put, so if we look at the CNN and the GCN, if we look at native structures, then we get this orange curve. So this is the recall versus precision. If we look at predicted, let's see, if we look at Rosetta predictions, we do somewhat worse. And if we look at David Jones positions or deep folds positions, which also I guess use Rosetta, then we do something in between. But overall, we're beginning to figure out how to use protein predictions to make these function predictions. And the orange curve is the beginning of showing us that de novo structure predictions are good enough to get improvement in function prediction. And the thing that we do here is we take the Rosetta structure prediction and the native for that protein. And when we put them through in the first layer of the graph convolution, we get some relationship between the features generated from the true structure and the prediction. As we get to the third layer, then the correlation between the prediction features and the native structures features actually get much closer. And so what this is showing us is as we go deeper in the graph convolutional neural network, layer 1, 2, 3 of the graph convolution, and there are only 3 layers, we actually have a much tighter relationship between the native structures features and the features of from the Rosetta prediction. And this is important because we want to predict the function of everything, and we only have the structures for 50,000 non-redundant sequences. So we need to use de novo and homology modeling in order to do that. All right, the last thing that I want to show you is I think the ultimate goal, which is we don't want to predict function and just produce a label for each gene. We actually want to highlight the residues, the positions in each gene that we think are responsible for the function. We want to sort these things out and generate testable hypotheses. And in large multi-domain proteins, that means nailing where the function is, right? All right. So in the early days of neural networks, I think people often referred to them as black boxes. And so now I think we have a lot of tools to figure out not what's going on in the black box, but what's going on in your feature space. And so in the end, there's a lot of parameters in any of these models. And so this class activation mapping or salience mapping, they're two alternates to the same idea, which is if you make a prediction. In this particular case, this was, vision people like to predict if things are cats, dogs, babies, they want to make their talks have, jokes built in. And so cats and dogs are great for that. So this is a prediction from this first class activation map paper where they say, if you make a positive prediction of this type of dog, then vary the features or vary the prediction. So either map in that way or that way to figure out which parts of the features when modified have some change or create some change in the prediction. So we want to find weights over all the set of features that, given a single trained model, tell us the change in the output as a function of the change in the input space. And so from that, we can actually come up with, for different layers in the network, how changing this last layer affects this prediction. We can propagate that back through the model to eventually produce the class activation map that identifies the dog here, right? There's been a lot of work to show that you can't just do this with a single model, but you need to train ensembles of models if you want robust class activation mapping. There's a lot of work that's gone on here to show that neural networks find the simplest rules if there are multiple entities in an input space that could produce that positive prediction, that if you want to identify all of them, you have to do additional things. So there's a lot of work in class activation mapping that we had to account for. And the main thing that we do is that we train 100 different graph convolutional neural networks, and all of the class activation mapping I'll show you is from an ensemble approach to this. So Vlad has been doing a lot of work to come up with new or robust class activation mapping. So this is what we get for proteins. And so this is a case where we put in this sequence. These are all examples from the protein data bank where we hid these proteins and all their homologs from the training and test sets. Then we get a prediction. In this case, we got a prediction, calcium ion binding. And that prediction is correct. This protein, if you look in the crystal structure, does have two calciums that are bound. If we then look at this class activation map, we get a few peaks, but we get two peaks here and here. And if we look at these residues here that correspond to these hot regions in the class activation map here and here, we actually light up all the residues that chelate that particular calcium ion. If we look in another protein that again is a proper prediction, we've simulated a temporal holdout to make this prediction. We actually get most of the binding residues correct with the class activation map. Vlad went a little crazy on calcium, zinc binding, RNA binding. We also see again larger binding sites, more complexity, but we basically light up most of the key interface with the RNA, and then we have some mysterious error down here. So we do make mistakes here. This is a case where this protein, again, this entire sequence was fed in without knowing what the domain structure was, and the class activation map lights up the helices that intercalate into the major groove there. Again, I'm showing you things where binding is in the word, is in the function label, because those are things that we can validate very, very, very specifically. This is glutathione transferase activity, and the activation map shows us the interface that glutathione sits on. including some of the catalytic residues that are over here. GTP, this one's a bit of a monster. Is this an error, or is this some structural component that we need to actually carry out the function of using this molecule to make an energetically uphill change in conformation? Who knows? This is an example that tells us that there's a lot left to investigate in terms of how to actually fit these models. We don't just want to fit these models so that they optimize predictive performance, but we also want to think a little bit about this being the eventual goal. All right, so this goes on, and I should probably save some time for questions. I talked today about the work of Julia, Vlad, and Doug, and their PhD underlings. So these guys, Emily is now at Epistemic AI. Meet just joined the PhD program at CDS. Doug, Vlad, and Julia are all fellows at the Flatiron Institute. This is the work that I talked about today, but we're applying this to these very large collections of proteins from the microbiome. So there's a whole facet to this program that involves where we get the sequences that we're predicting and how we prioritize them. And so Tommy and Tomas from Rob Knight's group and Romnick's group, and also IBM, also had a really big role in starting this project. So, and that's it for now. Thank you.